Hi everyone, welcome to Professor Long's Lectures in Anatomy and Physiology. I'm Professor Bob Long. This series of videos is intended for use by students who are enrolled in my Human Anatomy and Physiology courses at Del Mar College. If anyone else out there in YouTube land finds these helpful, great, use them. Otherwise, they're really designed for my students, how I test, how I teach, and what I think they need to know. And as you know, we're in the coronavirus shutdown, so I'm a face-to-face guy trying to teach online, so this is my method of delivery. So, they're done crudely. If you hear any barking or in the background from my dog or my kids, because of the shutdown, I'm having to do all this in my dining room. So, please bear with me. All right, this particular video is intended for my students in my Human Anatomy and Physiology 2 course. We're doing a series of visual physiology videos. Hopefully you just watched the video on the nature of light and what pigments are in the visible light spectrum. At the end of the video, I made reference to the fact that we're going to talk about the difference between what are called rods and cones. So if you're following along in my class, we're going to be on the bottom of page 15 or near the bottom where it says draw and describe a rod and cone. So the cells, the neurons, there's a specialized group of neurons in our eyeball, in our retina of our eye, called photoreceptors. So we still haven't really started talking about the neural tunic. We're going to, but I'm setting up for that. So the cells in our eyes, the neurons in our eyes that detect photons of light are called photoreceptors because they receive photons of light. Photoreceptors contain the visual pigments. And we just talked about the visual pigments in the last lecture, okay? Those substances that absorb photons of light within the visible spectrum. I know I'm squeezed over there, forgive me, okay? Now, there are two major classes of photoreceptors. And this is my abbreviation for receptor, so I'm going to make photoreceptor that way, just for shorthand. One of those types of cells is called a rod, and the other one is called a cone. Okay? We're going to draw and describe rods and cones. And I'm going to draw them here, in this space. Before I do, I want to give you some concept as to what some of the terminology I'm going to be using is. So now, if I drew an eyeball here, this would be the front of the eye where the cornea is, and then the back of the eye where the sclera would go back towards the optic nerve. The optic nerve comes in a little bit inferiorly here. As we know, that would be our fibrous tunic. And then we have a layer of blood vessels called the vascular tunic that comes into the eye and makes up a layer of the choroid. It has the ciliary muscle and the suspensory ligaments, and it also makes up the retina. I'm just going to use all red for our vascular tunic, okay? Not the retina, it makes up the iris and pupil. Now, just deep to all of that, there's going to be a dark layer of tissue that's going to cover just deep to the choroid layer. It's going to go all the way around here. It is going to run on the back of the iris, and it actually is going to cover the back of the ciliary muscle as well. That dark layer is going to be called a pigmented epithelium. We're going to define what the pigmented epithelium is after we talk about rods and cones, okay? Now, inside of that, there's a layer called the retina. And the rods and cones are going to be lying inside the retina. The retina is going to cover the inside of the eyeball here, like this, but it stops short of the front of the eye, and that would be our ora serrata lining the inside of the eye. The retina would be like the paint on these walls here lining the inside. Now, the reason the retina stops short at the ora serrata and doesn't cover all of the inside of the eye like the pigmented epithelium does is because of the openings where the cornea and the pupil are, no light coming into the eye at any crazy angle is going to land anywhere more forward than this spot. Oops, I didn't draw that very well. So why would I put photoreceptors where no photons can land? So Mother Nature's efficient, so we don't put photoreceptors where we don't need them. And so the ora serrata is the end of the retina where there's no more photoreceptors, no more retina. But the pigmented epithelium continues on to the back of the ciliary muscle and the iris. Now, these cells, since this is outside the eye and this is inside the eye, in my drawing, this would be outside the eye, this would be inside. So we're literally cutting sort of this and looking at it. When I look at a rod, Rods are funny shaped bipolar neurons. When you studied part 1 A&P, you know that we had neurons that had many dendrites called multipolar neurons and a single axon where they would synapse and release neurotransmitter. We also had bipolar neurons that has the soma in the middle, like this, and we had unipolar neurons. Unipolar neurons are usually incoming sensory neurons in the spinal cord. Well, since these would be the dendrites and this would be the axon, it turns out that photoreceptors are really just kind of modified neurons. They have dendrites and an axon, but the dendrites don't look right. And it turns out that the dendrites are going to be facing towards the outside of the eye, facing the back wall. If I draw this neuron, this would be the axon. Then we would have a soma with the nucleus and organelles and perikaryon. And then I have this long rod-shaped or cylindrically shaped segment. Since this is facing outside, this is called the outer segment of the rod cell. And about where the somata would be, the cell bodies, this would be called the inner segment of our rod cell. And the reason they're called a rod is because they look like a rod out here. They're cylindrically shaped. The outer segment of the rod is facing the back wall of the eye, sort of facing the pigmented epithelium, and it is a long cylindrical structure that is filled with little discs of membrane that are stacked in here. Now, I don't know how many of y'all know this unless you maybe work in a bank or in a business, but when I was a kid and we would get money, we would put little rolls of coins together, like a little stack of nickels rolled up in a paper roll. So when I look at a rod, the outer segment of rod is filled with little discs that are made up of membrane, similar to the cell membrane. If I took one of these discs out, they're little disc-shaped structures, almost like a little hockey puck. And in that disc, in the membrane of the disc, are what we call the visual pigments. And that would be the visual pigment that we described in the last video called rhodopsin. Now I know I used a red color, but rhodopsin does not absorb red photons. And so all of these visual pigments are literally staggered in here, thousands of them in these discs. So that if a photon of light tries to penetrate past this cell, the likelihood it's going to get through this cell, if it's within the visible spectrum, and miss all the pigments is pretty low. Okay? Now, just in case it does, we don't want the photon of light to bounce off the back of the eyeball, then bounce back out. Imagine if I shot a tennis ball at a really high rate of speed. It would be bouncing off the walls in this room, and every time it hits something, it loses a little bit of energy because the walls would absorb some of it. It would slow down, and then eventually it would stop rolling around the room, or start rolling around the room. Well, I don't want a photon of light that is outside the visible spectrum to bounce off the back wall, bounce off the back of the iris, and then slow down and then trigger vision. And there's a reason for that. I'm going to use a different color, okay? If I shot a photon of light in here, let's say it's ultraviolet light, I'm going to use purple. But if I shot some UV light in here, it's at such a high velocity that as it comes into the eye, if it bounces and bounces and bounces and bounces and then gets absorbed over here, as it loses energy, it then triggers visual physiology. I wouldn't see the photon of light as coming from over here. My eyeball would think that it's coming from somewhere over here a few seconds after I actually could have seen it. So when a photon of light hits a photoreceptor, we want it to trigger that photoreceptor or get absorbed. Well, that's what the pigmented epithelium does. This layer here is going to be a layer that we call the pigmented epithelium. I'm going to put PE there for pigmented epithelium. If the photon of light is not absorbed by the photoreceptor, it will get absorbed by the pigmented epithelium. Another thing the pigmented epithelium does is this. As we trigger these visual pigments, when they get hit, there's a part of them that is made out of protein. When they get hit with the photon of light, they change shape. And as the photon of light is absorbed, eventually they will reset so the next photon can cause them. And every time they get hit, they trigger and then reset. Well, just like anything, if you fold it and unfold it and fold it and unfold it, it will start to break apart, even a piece of metal. So the visual pigments can wear out over time. The newer ones that are manufactured are closer to the soma. The older ones get pushed out. As they wear out, we can actually shed those discs and the visual pigments, and they get absorbed by the pigmented epithelium. And so the pigmented epithelium is filled with visual pigments as well. But it does not trigger phototransduction since it's not a neuron. So now, when we describe a rod, this is how we describe them verbally. Rods have an elongated or cylindrical outer segment filled with numerous membranous discs that contain the visual pigment rhodopsin. Again, rods are photoreceptors that have elongated or cylindrical outer segments filled with numerous membranous discs, and those discs contain the visual pigment rhodopsin. And one of the things we know about rods, I described in the last video, that rhodopsin is active in a dark room or in dim light, and they do not allow us to see any color. They are black and white vision or shades of gray. Now, when we describe a cone is also going to have an inner segment, which is an axon, a soma with a nucleus, and their outer segments are actually tapered or conical in shape like this. Hence the name a cone. They are also filled with these little membranous discs, although some books will say that they have folds in the cell membrane, but there's all these little membranous discs, and the discs can get shed and they get absorbed by the pigmented epithelium as well. And on these discs are the visual pigments. If I pulled one of these discs out, what I would see is on this disc, instead of rhodopsin, I might have one of the other visual pigments. If this one makes the visual pigment to absorb those photons in the blue part of the spectrum, then we would say that this would be iodopsin 3, and this cone is sensitive to blue light, or we sometimes refer to it as a blue cone because they're sensitive to photons in the blue part of the spectrum. If I had another cone that made iodopsin 1, then it would only make the visual pigment iodopsin 1 and only be absorbing photons in the red part of the spectrum, so it would be called a red cone. If it made iodopsin 2, it would absorb green light. So we have 3 distinct populations of cones. So before I go there, let me just say this. When we describe a rod, we say they have an elongated or cylindrical outer segment filled with membranous discs that contain the visual pigment rhodopsin. Cones have a conical or tapered outer segment filled with membranous discs that contain the visual pigments iodopsin 1, iodopsin 2, or iodopsin 3. If a cone makes iodopsin 1, it can only make iodopsin 1 and absorb in the red part of the spectrum. If a cone makes iodopsin 2, it can only absorb in the green part of the spectrum, and it only makes iodopsin 2. And if it makes iodopsin 3, then it's a blue cone. It can only absorb in the blue part of the spectrum. So now on the bottom of page 15, we've drawn and described the rods and cones. Elongated or cylindrical outer segment, visual pigments in the discs, tapered or conical outer segments with membranous discs that contain the visual pigments iodopsin 1, 2, or 3. Again, a cone can only make one type of visual pigment, either iodopsin 1 or iodopsin 2 or iodopsin 3. They can't make more than one. So the information I'm covering here is on the bottom of page 15, and it is all explained on page 16 in this little paragraph. So make sure you read that and you understand it. Okay. So now, we have this idea of what rods and cones look like. We have this idea that they're facing the back of the eyeball. Now we're going to go to the very beginning of the neural tunic. So we're going to jump back to page 14 for a moment, and we're going to go in the order of the note set finally. All right, so we're going to be doing page 14 where it says the neural tunic in my note set. Now, I hope you got this. I hope you've drawn it or have a good vision of it or an idea of it in your head. I'm going to erase it so that we can cover all of this information. All right. So the first thing about the neural tunic that I want you guys to understand is that the neural tunic is the third of the 3 coats of the eyeball. Okay, we said that the eye has 3 tunics. That's where we started our visual physiology. The first one we said was the fibrous tunic. We said the fibrous tunic is dense connective tissue mostly. and it includes mostly the sclera and the cornea. The second tunic we said was the vascular tunic. The vascular tunic allows blood flow into the eye and has a lot of blood vessels, but it has a number of structures. It includes the choroid layer, it includes the ciliary body, it includes the iris and pupil. We've talked all about that. Now we're doing the third tunic of the eye called the neural tunic, because the neural tunic contains the neurons. The neural tunic is made up of two structures. One of the structures it's made out of is called the pigmented epithelium. I talked about this in the last video, but I want to repeat it. Sometimes I repeat things on purpose because I want you to hear it over and over and over and over again till it sticks into your brain. Okay, so the pigmented epithelium is a layer of cells in the retina, we should say, containing pigments that can absorb photons of light that are not absorbed by the retina. Okay? We can say that the pigmented epithelium is a layer of cells that contains pigments that can absorb the photons of light that are not absorbed by the retina or by the photoreceptors. And sometimes we say that it absorbs those photons of light that are outside the visible spectrum because it can absorb some of those. All right? So now, the second layer of the neural tunic is the retina. The retina contains 3 layers of neurons that send action potentials out of the optic nerve. Okay? That's not really the best way to define it, but the retina has 3 layers of neurons. One of those layers of neurons is going to be the photoreceptors that receive the photons of light, absorb their energy. It will change their resting membrane potential and alter their physiology, and then they're going to signal 2 other layers of cells that will eventually send action potentials out the optic nerve. Okay, I know my penmanship is not the best, but I'm trying my best. All right. So now let's talk about retinal organization. Okay. So on page 14, it asks you to define diabetic retinopathy. We might talk about that, but ultimately I think you should look that up. As a matter of fact, I'm going to write this down for a second, and then I'm going to move on to the next page. Okay. So now, when it comes to the vascular tunic, it's pigmented epithelium that absorbs photons of light not absorbed by the photoreceptors or those outside the visible spectrum. The retina contains the photoreceptors that absorb the photons of light in the visible spectrum, and 2 other layers of neurons that transmit the signal to our brain. So now, when it comes to what we call the retinal organization, actually, let me talk about diabetic retinopathy real quick. Diabetic retinopathy. You can look this up. I want you to look it up. But it's a pathology of the retina that is associated with diabetes. Now, in diabetes, we don't have the ability to process sugar properly to turn it into energy in our mitochondria. We can't get the sugar into the cells. Neurons are the biggest wimps of the human body, and if I cut off their sugar supply or their oxygen supply, they start dying off rather rapidly. So in diabetic retinopathy, the photoreceptors in the eyeball are dying because they can't get their sugar. It's a pathology of the retina due to diabetes. Now, our blood vessels, our blood cells, have smooth muscle in them. It utilize calcium to contract, if you remember muscle contractions in part 1 A&P. Those little calcium channels allow calcium to flow in, and calcium pumps pump the calcium out. We need energy from sugar to pump calcium out of our smooth muscle in our blood vessels, in the walls of a blood vessel. Without the sugar, the calcium stays trapped in the muscle cells and can start to crystallize or calcify. And as they calcify, the blood vessel will eventually get clogged off, almost like calcium salts in our water lines, clogging off our water, decreasing the diameter, and eventually it can clog up a water pipe. So In diabetic retinopathy, the calcium channels can't function and the smooth muscle will start to harden and crystallize. And the blood vessel can get constricted. So if you look inside the retina, there's a structure called the fovea, and we have the optic disc, if you remember from the models in lab. And the blood vessels enter the optic disc. As the blood vessels enter the optic disc, they will fan out in a specific pattern. And that pattern is unique to each individual, but there's a certain pattern that should be there. If these blood vessels get clogged with calcium, then they won't show up. They die off. And so you get what's called, when you get a lack of blood flow, that kills the neurons. Our body, in a last-ditch effort to try to sort of save those cells, will start growing all these little branches of blood vessels around that blockage. And so you get what's called a neovascularization. Neo meaning new, vascularization meaning forming blood vessels. So sometimes your optometrist or ophthalmologist can look in your eye and go, hey, have you been diagnosed with diabetes? They see some diabetic retinopathy. Anyway, and neovascularization. You need to know this definition, you need to know that definition. Look them both up, okay? Now in the note set, we're going to switch over to page 15, and we're going to draw and label the organization of the retina, okay? And this is important for our understanding of visual physiology. So, if I were looking in the eyeball, I'm going to redraw this again. Here's the cornea, here's the sclera, the front of the eye, and the back of the eye. As you know, my photoreceptors, I'm going to make a little blue cone-shaped cell here. My cone is going to be facing in this direction. Obviously, they're not this thick, but I'm over-magnifying. The drawing is not to scale. All of these cells would be squeezed into a very, very thin layer. But I'm going to have my photoreceptor facing outward that way, okay? So if I were drawing this, here would be my cone cell. I'm just going to enlarge this drawing like this, okay? Now the cone cells are going to be facing the pigmented epithelium, which is right here. So I'm going to have my pigmented epithelium. Again, I'm just going to put PE here. I'm going to put photoreceptor here so that we can see the layers in order. Now right beyond the pigmented epithelium is going to be my choroid layer, my blood vessels. And then outside of that is going to be sclera. Since the sclera is filled with collagen fibers, I'll make it pink because collagen stains pink on our slides, and it's going to be a very thick layer. This almost is going to look like the slide of the retina that we do in lab. So this would be the sclera. The red would be our choroid. And then I have pigmented epithelium. And then I have my photoreceptors. And the nucleus of the photoreceptor would be here in the soma. And again, there would be discs out here with pigments. Now the next cell in the row is going to be a cell that looks like a classical bipolar neuron here, with the dendrite and the axon. So this layer is called the bipolar cells. The third layer of cells are going to be these short, funny looking cells that send axons out the optic nerve. And so these cells are referred to as a ganglion. Remember, a ganglion is a collection of neuron cell bodies outside the CNS. So I would have a bipolar cell here, and then I would have a ganglion cell send their axons out. And if I had another series of 3 cells, let's say I have a rod here, then a bipolar cell, then a ganglion cell, all the ganglion cells, no matter where they're at in the retina, send their axons out the optic nerve to our eyeball. Now this is kind of counterintuitive to what you would think. You would think since a photon of light is going to come in the cornea, it's going to pass through the retina, through the lens, and then be focused on the retina. You would think that the photoreceptors would be the cells that are facing where the photons of light are coming in, because the photon of light is going to come in this way. This is our retinal organization. The photoreceptors are up against the wall. The pigmented epithelium. The photoreceptors are facing outside. Then we have bipolar cells and ganglion cells. Okay, it's backwards. If I ask 3 of my students to line up and I'm going to throw a tennis ball, if you catch 100 of the tennis balls that I throw, you get a point. You would think that the first person who has to be up against the wall, the next person standing in front of them, and the next person standing in front of them. If they catch it, one person kicks the other, that person kicks the next person, and then the third person can stomp on something on the floor that lights up on the wall so I can see where the tennis ball was caught. You would think that it would be the person facing me would be the photoreceptor. But imagine if the person facing me is up against the wall, and then there's someone standing next to them or in front of them and someone else standing in front of them, and then they step on a button to tell me if they caught it. When I throw a tennis ball, the person who's the furthest away from me catches it, then signals the next person, which signals the next person, and they tell my brain. So a photon of light is going to enter our eyeball. And if that photon of light lies outside the visible spectrum, so if I do a photon that is outside the visible spectrum, one that cannot be absorbed by our photoreceptors, that photon of light will come in, it will pass the ganglion cell, it will pass the bipolar cell, it will pass the photoreceptor, and then will be absorbed by the pigmented epithelium. That's the order of structures that a photon of light would pass into as it's absorbed. Okay? I like to ask that question sometimes. Please list the structures in order that a photon of light that lies outside the visible spectrum will pass into the eyeball. If that photon of light triggers vision, let's say it's a blue photon, then when that photon of light comes in the eyeball, and if this is one that's in the visible spectrum, or what we'll say is a visible photon, a photon of light that lies within the visible spectrum, it will come into the eye and get absorbed by the photoreceptor and trigger it. It turns out that when we're in a dark room, photoreceptors are tonic receptors. They're always releasing neurotransmitter telling this bipolar cell we're blind. When they absorb a photon of light, there's a change in resting membrane potential. There's a series of physiological steps similar to olfaction, where we amplify the signal with adenylate cyclase and cyclic AMP. It's a different series of steps. We have phosphodiesterase and transducin, and I'm not going to get into the steps. But let's say that in a dark room with no photons, this photoreceptor is releasing neurotransmitter telling this cell we can't see anything. When the photon of light is absorbed by the photoreceptor, it stops releasing neurotransmitter. Then the bipolar cell fires an action potential, telling the ganglion cell that it's going to fire an action potential, and then we send the signal out of our optic nerve to our brain. So let me repeat this. When I have a photon of light that lies outside the visible spectrum, what would be the order of structures that it passes as it's absorbed? It would pass by the ganglion cell, the bipolar cell, the photoreceptor, and is absorbed by the pigmented epithelium. If a photon of light lies within the visible spectrum, what would be the order of structures that fire action potentials or fire a signal telling our brain we're seeing? First, the photoreceptor fires, or actually it stops releasing neurotransmitter, but it fires. The second cell is the bipolar cell, then the ganglion cell is triggered, and then we send an action potential out the optic nerve to our brain. So this is our retinal organization that you should know, and that would be the order of cells that photons of light will either pass by if they're not inside the visible spectrum, or the order of cells that are triggered to fire if it lies within the visible spectrum. Now I'm going to cut the video here because we got to go into some other details of vision, but we're just about done. As a matter of fact, before I do that, let me make sure I want to stop, okay? I'm actually making up these videos as we go along, so I don't really have them planned out in advance, just like when I walk into class. So let me do one last thing with you guys, and then we'll actually be done with vision, all right? So there's 2 questions that are on page 15 of the note set that I want you guys to understand right after this retinal organization. So one of them is, what is the fovea centralis and what is the optic disc? So I'm going to erase all these notes and I'm going to talk to you about what the fovea is, and a little bit more about the organization of the retina. Okay. Now, I wish I didn't erase that eyeball there, but I'm going to redraw it anyway, so it'll be neater and cleaner. Okay. So here's your cornea, here's the sclera, and as you know, the optic nerve comes off a little bit low and downward. Actually, they point medially also. We know we have the lens here with the ciliary muscle and the suspensory ligaments and the iris and the pupil. I'm not interested in that. I'm interested in the retina itself. Now, we know that the optic disc is a spot on the retina. I'm going to define the optic disc first because it's easier. The optic disc is a spot on the retina. It's a disc-shaped spot on the retina where all axons of ganglion cells exit the eye via the optic nerve. And there's actually no photoreceptors on the optic disc. So if a photon of light lands on the optic disc, we're actually blind to it. We have a blind spot in each eye. And by the way, just so you know, both of our eyes are not focused exactly on the same point. They're off just a little bit, which gives us a three-dimensional awareness of the world. We don't see a flat world, we see a 3D world. But the optic disc doesn't have any photoreceptors on it. So each eyeball has a blind spot. As a matter of fact, there's a really cool thing in your textbook somewhere. I don't know where it is, but there's a picture in your lab manual or your textbook that has a cross and a dot. If you stare at the cross, close your left eye and stare at one object and move the book, eventually the other object disappears because the photons of light are landing on your optic disc. So you can see the blind spot, or you can experience it in each eyeball by following that little experiment. Now, the fovea, when I look at the retina, if I could cut the eyeball in half this way and look straight into the eye, so I'm looking at the retina as a big disc, then the optic disc is a little bit inferior and medial, but there's a spot right in the center of the visual field called the macula. You may have heard of macular degeneration. That's when the neurons in the macula are dying, usually in diabetic retinopathy. Within the macula, there's a smaller spot inside of here called the fovea, also called the fovea centralis, because it's in the middle of the macula, or the central part of the macula. Now, one of the things I want to show you is I'm going to redraw this next to it for a reason. If I'm looking at the retina, when we're staring at an object, most of the photons are coming straight in the eye and they're going to hit this area. And the ones that are spreading out might land in other parts of the eye, but some of them are blocked. But an overwhelming majority of the photons of light, if I'm staring at a particular object, are going to land right in a specific spot, which should be right in the center of our visual field. It turns out that if I start at that spot, I'm going to use red as my color for cones.